developed for shellfish. But I'm going to make some comments about the benthic processes and models at the beginning here. But going through these models, they were developed for a benthic organism. But I think maybe I'd like to look at this in terms of a more general way in which models are evolving, and it goes to some of the comments we've heard this morning about how we go from these simple parameterizations, empirically based models in effect, to models that have more of a mechanistic basis to them. All right, so I am also going to talk a little bit about, in the context of these models, human impacts and management applications. And this isn't at the level of something like Atlantis or these end-to-end models. This is a specific application to look at a specific problem. And models are good for that. You don't need the full suite of everything in order to address an issue that may have relevance to society. Okay, so what I want to talk about here in the presentation, first talk a little bit about the benthic invertebrates, look at a population-based model, talk about the approach to that, modeling these benthic shellfish populations, and then the limitations of these approaches. Then a little bit about our individual-based model, the approach, combining it with the population model, and then genetic variability. All right? And Barish mentioned the fact that we're getting a lot of the genomic information. We're learning a lot about the genetics of organisms now. And if you really want to attribute cause and effect, you really have to get down at the genetics level. Also, it's through the genetic adaptation, the genetic evolution, that we're going to evolve or change in response to climate change, things like increasing pH, warming temperatures, and things like that. So we have to be able to deal with that somehow in the context of these models. And then just including remarks here. Okay, so benthic invertebrates, they're important parts of estuary and coastal food webs. They're important in general. A lot of them are chosen as sentinel species because they're long-lived. That's why we focus on shellfish in a lot of environments because of that. They provide coupling between the benthic and pelagic systems. Most of these, like the shellfish I'm going to talk about, have a post-settlement phase, which is sedentary, which is why they're a good sentinel species. And they also have a larval phase, which connects back to the pelagic system. All right. They filter water. They're important in an ecosystem services way because of filtering water in bays and estuaries and coastal environments. And through filtering, they do things like regulate food concentrations. They actually can control the distribution of phytoplankton in areas. And they're also important in terms of nutrient cycling and returning nutrients to the water column. The other thing that's becoming increasingly people are aware of right now, things like shellfish are important to carbonate balances of estuaries. Right now, almost every estuary in the world has a negative carbonate balance, which means there's not enough shell in the estuaries, because when people harvest shellfish, you don't put the carbonate back in. All right, so I live in an area where the oyster shells and the clam shells are ground up and turned into sidewalks. All right, so if you're walking on shellfish sidewalks, that means your local estuary doesn't have those shells anymore. So this is becoming a problem with restoration efforts in trying to restore these estuaries to the previous forms. All right, and then the other thing is that many of these benthic invertebrates are commercially important species, and they're important to local history and culture. And I think Beth mentioned that earlier, or yesterday in her presentation, that a lot of this is cultural, what we value as a culture, and that's why we want to restore it, we want to save it, and maintain it. All right, so benthic invertebrates, if you look through the literature and you look at the models that have been done, usually for coastal and estuarine systems, and you look at the kind of model that Barish was just talking about, the NPZ types of models, there's usually a bottom boundary condition in areas where the bottom is shallow enough that it matters. All right, and the benthic invertebrates are parameterized in those models typically as a bottom boundary condition, usually in terms of nutrient supply back to the water column or through some filtration sink that just sucks things out of the water column. All right, so it's a loss of phytoplankton or nutrients. right? And, or there may be a general benthic population that sort of grows and declines in response to various environmental conditions, such as, bottom temperature, sedimentation rates, that kind of things. All right, the other type of model that's looked at sometimes is a coupled circulation biological model that usually focuses on the larval part of the animal's life history. So where do they go, where do they settle, and that's important in terms of maintaining populations, in terms of management. If you want to restore this reef, are you restoring the reef that is actually providing a source of larvae for the rest of the estuary, or is it the sink population, that kind of thing. And then there are some models that go all the way from circulation biology that includes the larvae and the post-settlement populations. And some of these are actually coupled to lower trophic level models. So there's a dynamically evolving pelagic system with a dynamically evolving food field, and you've got a real population on the bottom that's eating that and then contributing back through nutrients. So the message here is that there is there are a number of ways to do this, and a number of ways to put the benthic environment into these various kinds of models. All right, and all of them, though, and what I hope to bring home as a message today, is if you're going to do this, you first have to understand the ecology, the physiology, and the life history of the organisms or organism that you're dealing with in terms of the benthic environment. And that goes to all of these. I think that was Barish's comment and various other comments we've heard from other people, is that I don't think anymore you can get away with just having the average phytoplankton or the average zooplankton. We're recognizing that species matter. And so that's why the emphasis now on actually understanding the life history and the biology of these species. Okay, a couple more general comments before I get into the slides here, into the models. Most models, the models I'm going to talk about, and even the majority of them, track energy, usually in terms of calories, joules, or sometimes in terms of carbon. And for these species, that energy is given by the difference between assimilation and respiration. So what's left over is what goes into the animal. All right. Now, when you start dealing with things at the level of zooplankton and higher trophic levels, you have to deal with reproduction. It's particularly important when, like with these shellfish I'm going to talk about here. So you also have to not only grow the somatic tissue that the animal needs to maintain itself, you also have to produce the reproductive tissue along with it. All right. There's also mortality. It's natural mortality, predation mortality, and things like starvation that comes from lack of good quality food in many cases. The other one that was just mentioned is disease. All right, and this is a major problem right now in a lot of the estuarine and coastal systems, the shellfish and other types of invertebrates, a lot of, again, commercially important species are being impacted by disease. So there's a lot of work going on right now to understand that and understand disease transmission in marine systems, because the models we have for how we transmit diseases among ourselves don't work in marine systems. Those are density-based contact models. They don't really work for marine populations, and that's a whole other lecture I could give. But it's important, and I'll just put that out there as something to think about. Okay, there's also the pelagic phase of the life history. You need a circulation model, you start dealing with that. And then there are ranges of responses within species in their rates, like their respiration rate, assimilation rate. We don't all respire or assimilate at the same rate, and that's the result of genetic variability, right? And that's something we want to build into these models now, because that variability is what gives the adaptation to changing environmental conditions. All right, and I'm going to, there are multiple kinds of species. Typically the models focus on the ones for which we have the data and the understanding, and that's what I'm going to do here is to talk about some oyster and clam models. Okay, now, if you have a benthic oyster population and you want to look at the effect of that as it grows and dies and that kind of thing, most of the early models, the population dynamics models for these kinds of organisms in the past, were population-based models of the average individual. All right, and it was this kind of flow diagram up here where you filtered the animal the average animal population filters water, that goes to ingestion, assimilation here. You debit that with respiration, and you end up with net production here. Right? That's a fairly standard approach in all of these models. All right, but in this type of approach with the general population model, that's all parameterized from whatever the average individual would be, which we've heard several people say the average doesn't exist. Okay. All right, this type of model, you can put salinity, temperature, and the food field in there, and so you input those as variables, the animal responds, the population responds to it, and you get solutions or simulations from that. Okay. Now, that kind of model is typically a population model. You start out with, say, the early stage, the newly set juveniles, and you grow it up to the adult. All right, those are it's basically size class-based models. All right, so as the animal grows, it produces somatic tissue, and then also as it gets older, it forms reproductive tissue. And then the reproduction occurs in response to biological or environmental cues of some type. The animal filtration rate sets up this assimilation and respiration, and in fact that's important. If you key in on what needs to really be parameterized, that's where you look. All right, now when you start dealing with things much above the level of zooplankton, you find out that the animals gain and lose mass. All right, so they starve, they actually shrink. Right? And that needs to be built into these kinds of models, is that this net production here gets divided between somatic growth and reproductive tissue, but if the food field or the temperature conditions are bad for the animal, it can't grow and it will actually shrink. All right, and these models are usually in terms, as I said, of energy or carbon units. And because they are size-based models, one of the things you have to end up doing is developing some type of length to weight relationship. The biological literature is full of these kinds of relationships. This is the same species oyster in different parts of an estuary. You don't get the same length weight relationship. Usually you take these, draw a line through the middle of it, and that's your length weight relationship for these models. All right, this is one of the, this is a scale over here where this has been now just turned into discrete size classes, and in terms of the biomass and the length of the animal. All right, so it's a nonlinear size scale that you use to go between these different size classes. We'll come back to the implications of that here in a moment. All right, so if you look at the model, let's exclude the the physical environment, the circulation, and everything. You end up with just sort of ordinary differential equations. All right, so net production ends up being somatic tissue, reproductive tissue. It comes from the difference between assimilation and respiration for a particular size class. So now you've got just a time rate change in the oyster here, given by its growth in the somatic and reproductive tissue. All right, but if you're at size class J, you get individuals moving up from the lower size class, and then this one grows and moves up into the next. So you gain from below and you lose to the bigger one. All right, so that's what that is, and that gives you then your time change in that particular size class. All right, now, size class models, right, when you start out with something like this, you start out with, say, 10 animals per meter squared. Right? At the end of your simulation, you still need to have 10 animals per meter squared if you don't put in new recruits. You just grow those animals. All right, now, if you have 10 units of energy formed in size class J, and you move it up to size class J plus 1, that 10 units isn't necessarily one full animal, okay? All right, so what you have to do is you scale the transfers above it and below it by the weight of the animal. All right, now I mention that because if you go back and look in the literature at the early models that were done like this, they weren't scaled. So in effect, what happens is you create destroyed animals by moving them up and down. And it sounds silly, but you really have to, not silly, but you have to conserve the number of animals. Because as far as we're shown, we don't have a lot of governing principles in ecological, biological model, but conservation of mass is one of those. All right, so you have to be careful with this to make sure that even if you're running your model in energy units or carbon units, you still have to conserve the number of animals that you have.
Eileen, and again, just to So the spawning and recruitment?
No, no spawning, no recruitment. Okay, this